When thinking about the size of the cell, the first thing that comes to mind is the cell needs to be small. These reasons are because when a cell gets bigger or smaller, the amount of DNA doesn't change, so the nucleus has a hard time distributing things. As you can see in the photo, the nucleus does not change its size, but the cell does. Also, another reason is it gives oxygen, water, and carbon dioxide a hard time to get into the cell because of its size. It's hard to distribute everything to the rest of the cell when the nucleus size does not change. Surface area and volume both grow when the size of the cell increases, but they don't grow at the same rate. Next is cell cycle. Cell cycle can be broken down into two major stages, interphase and mitosis. These can both be broken down into categories. First, mitosis can be broken into 5 stages: prophase, metaphase, anaphase, telophase, and cytokinesis. Interphase can be broken down into 3 stages: G1, S, and G2 phase. Interphase is where the cell spends most of its time. The G phases are the growth phases, and the S phase is where the DNA replicates. The reason the cell is in interphase the longest is because it needs to grow and develop. Mitosis is the dividing stage. The 4 stages of mitosis are interphase, prophase, metaphase, anaphase, telophase, and cytokinesis. Interphase is the growth and development stage. Prophase forms some sister chromatids, and they are freely moving around the cell. The central mirror is what connects the copies of chromosomes. In metaphase, the sister chromatids are going to move to the center of the cell. The centrioles will make the spindle fibers and will pull the chromatids apart. In anaphase, the chromosomes will be pulled apart by the spindle fibers. In telophase, the cells will regain its nuclear envelope, to go into interphase to do its process all over again. Next, I'll be talking about the difference between plant and animal cells in mitosis. The difference in animal cells is the membrane will be pulled apart. In plant cells, they will grow a cell wall in between the 2 new cells. As you can see in the photo, in the plant cell, it looks like they're connected but have a cell wall in between them, and with the animal cell, they don't. Next is cancer. Cancer can be caused from the cell cycle. The reason for this can be caused by your diet or smoking or mutation. Cancer can cause many issues like tissues losing its ability to control regulation and steal nutrients from other things. In the cell cycle, there are checkpoints that if the cell doesn't meet certain things, then it won't be able to keep moving on to each process. For solutions of cancer, you can get radiation or targeted therapy, chemotherapy, and many more. When talking about DNA structure, it is important to note that G and C go together and A and T go together. This was discovered by Chargaff, and he discovered that these DNA strands came together. Rosalind Franklin is another scientist to note because she discovered that DNA is a helix structure, and Watson and Crick found that it has a double helix pattern. Nucleotides have 3 things that are very important: phosphate group, sugar, and nitrogen base. These are put together by hydrogen bonds. This makes DNA replication easier. Next is DNA function. DNA has a few different functions. It codes for many proteins, which can lead to putting different DNA and combining them into organisms. The second function DNA has is it stores genetic material in the DNA. Lastly is DNA replication. 2 important things to note when talking about DNA replication is the DNA polymers and helicase. The polymers reads the DNA and adds new complementary bases. The helicase unzips the protein to get the double helix into one strand. This allows the new cell to have one new strand and one original strand. Thank you, Mr. Carlson, and hope you stay safe during these times.